Okay, we might kick off our next session, which is on bioprinting, art and science. I'm just going to introduce our two speakers now so that we can move through seamlessly after. Our first speaker is Svenja Kratz. She is a contemporary artist and a lecturer at the University of Tasmania. I'm very excited to see her presentation because people keep telling me how cool her artwork is. And our second speaker is Associate Professor Elizabeth Stevens, who has just started a future fellowship, which is also very exciting. I'm just going to tell you what it is about.
I'll tell you what it's about.
Yeah, why don't you?
Presentation, so you
Okay, let me just give a quick sentence because the Future Fellowship examines the cultural history of the experiment and contemporary practices of experimentation, from the emergence of 19th century scientific labs to contemporary experimental art. So that will also touch on the 3D printing aspects, which is definitely part of experimentation. So I will hand over to Svenja now.
Thank you very much, and thank you very much to Matthew as well for inviting me to come along today to speak. It's always wonderful to actually connect with an audience outside of just the arts and humanities. So thank you very much. Today, as you'll see from my slide here, I'll be talking a bit about one of my recent projects, but I'll give you also a little bit of a background of my practice, where it's come from, and also where it'll be going, and then it'll work nicely in terms of complementing the discussion of Elizabeth afterwards. So just to give you a little bit of an idea, I actually am a visual artist, but I've been working across art and science since about 2007 as part of my PhD, which I completed here at QUT in a creative partnership with the Creative Industries faculty and the Institute of Health and Biomedical Innovation. I was really fortunate to have Z Upton as my supervisor, so I was really integrated into the tissue repair and regeneration group. And that actually allowed me to go into the lab to get trained with using the biotechnologies and to experience firsthand how do you grow cells, how do you actually do tissue engineering, all of those things. And that really informed my artistic practice. So a lot of my work, so there you can see back when I had short hair as a first year PhD student, fascinated by looking through the microscope and observing cells. And I also got to do things, so cell and tissue culture, also some genetic engineering, just basic stuff, so introducing fluorescence into the cells with green and red proteins, fluorescent proteins from marine organisms. And I produced a range of work, so sculptural work, video work, across a whole range of different mediums. One of the projects, just to give you a bit of an idea of how the cell culture comes into it, was towards the immortalization of Kira and Rama. And this was a project where I actually went to the abattoir to get to fetal calves, and what I was really interested in was the use of fetal calf serum in the development of a lot of these technologies. So the way that you actually work with the serum, which is derived from the blood of fetal calves, as part of the nutrient supplement that you use for growing cells. I was really interested in, well, where do these calves come from? And as an inexperienced scientist, and not a scientist at all, I had these visions that they were actually being grown in like vat somewhere in a commercial sort of industry. But really what I found out was that a lot of the fetal calves are actually a byproduct from meat production. So what I was quite interested in was actually looking at the boundaries between life and death. So I went to the abattoir, got the fetal calves, isolated cells, and they'd been dead for about 2 days. And then I seeded them into a silkworm cocoon that was displayed as part of a sculptural bioreactor. So it was kind of looking at how, when we're working with biotechnology, it shifts our understanding because those bodies were dead and yet they were also partially alive. But also looking at how we might want to distance ourselves from a lot of the technologies, but at the same time we're implicated into them because of the processes that are already embedded. So these processes of consumption that we're often not aware of. So we're sort of looking at a range of those things. I also developed other work with tissue engineering and things like that, but that gives you a little bit of an idea. And the aim of the work that I was doing at IBI was really around the critical and creative affordances of biotechnologies. More recently, I've been lucky enough to work with the Centre for Regenerative Medicine with Professor Dietmar Hutmacher and a lot of his research team, exploring particularly the creative affordances, working with biofabrication. So this is a project that came out of a Creative Sparks grant from the Brisbane City Council, which was sort of a starting point for us to investigate opportunities for collaboration. Initially, I was interested in exploring some of the scaffold production techniques and to actually develop a basic perfusion bioreactor system. So that's where you actually push the media through the structure so you can actually feed it. But also have a holographic display system that would also use a kind of projection of the aliveness of the structure to kind of provoke empathy. So how do you actually provoke empathy for living products? That was sort of the initial framework that we were working from. Here you can see this is actually Tony Ausler, who's a very famous video artist, and that was kind of how I imagined this little entity might look. But as we sort of started working with those technologies, and I was also really inspired by a lot of the science fiction movies that were coming out at the time. So that's also where it was wonderful to see Matthew's talk, and where he was actually talking about Westworld and the way in which a lot of these contemporary sort of popular imagery really sort of start to interrogate a lot of the emerging technologies. And so Westworld, Black Mirror, all of these sorts of things also became really interesting to me. And then also what was actually playing out in terms of what's being pitched as a legitimate project. So here you've got the 2045 Avatar project. And this was a project that was initiated by Dmitry Itskov. And it's really kind of based on this staying alive, you know, immortality through engineering kind of approach, where he's really proposing, again, our biological bodies so imperfect, so terrible. We actually need to create a robotic body. We need to develop an artificial brain so that we can live forever. So you can see, again, really problematic assumptions that are playing themselves out here. You've got these problematic assumptions around also access, so who has access to these technologies that will be able to become immortal, but it also starts to reinforce a lot of these ideas around the mind-body dualism and the fact that, again, all of who we are, our consciousness is just in the brain. So if we can just somehow transport our brain into a robotic body, wouldn't that be a perfect system? And then eventually we could become a completely digital entity. So you can see here it goes all the way through, and the last avatar, Avatar D, in 2040 to 2045 is actually going to be a holographic projection of a body and a human. And that's how you'll achieve this immortality. So again, really problematic. But there are other things that are playing themselves out in that area of notions of immortality as well. Humai was another project that was looking at human resurrection through artificial intelligence. So again, this is a way of using artificial intelligence and nanotechnology to be able to preserve your identity so that, again, you could live forever. Again, this is one of those projects that sits sort of in that space where it could be more of a speculative account, and it's sort of, you know, is difficult to determine whether it's a serious project or operates more on the level of provocation. But then you've got these other projects, so Eternomy and then the Eternine, which are two projects that are talking about how they could essentially harvest information from your digital footprint and then recreate you and preserve you as a kind of digital entity that can continue to post in social media after you're dead and maintain that connection with your loved ones. So, I mean, again, really, really interesting, but also problematic projects. So along with this, what I kind of find interesting is, again, even in the sort of science realm, you've got this emphasis on the brain as being the center of consciousness, the center of identity. People like John Smart who are proposing, well, if we can just chemically preserve the brain with glutaraldehyde, which will just kind of bind proteins, later on we can resurrect you because we can scan those neural connections and then have your consciousness preserved, all right? A little bit like cryopreservation when that became popular. Sebastian Sung also believes that, you know, the root of our identity are just the neuronal connections that we have. So if you can, again, map the brain, you would be able to preserve consciousness. Of course, when you look at these proposals, it's again this really limited kind of reductionist perspective without taking into account of the complexity of the body. So the brain, of course, is connected into the whole nervous system. You know, you've also had Neumann Deutsch with his very popular book that talked about brain plasticity and the fact that our brains do change over time, and they change particularly through embodied interaction with the world. So we're not just this floating brain that kind of goes around that doesn't have any actual connection to the world around us, but it's very much about being part of a body, being embodied, and embodied kind of interaction with the world. And now you've got also more recent developments that talk about the relationship of hormones to brain function, and also the gut and the brain in the way that the flora inside your gut will also determine how your brain operates and things like that. So for me, I kind of was looking at all of this, and that was sort of a source of inspiration to kind of think about, well, what happens in this terrain, and also starting to think about immortality and the possibilities of that, and also this regard around how our bodies are seen as essentially a spare part that can just be incrementally replaced, and particularly with new technologies like biofabrication, where your heart fails, just print a new one, put it inside. Oh, you've lost a limb, that's okay, print a new one and just attach it and that's fine. Really you can just replace the entire body, and all you need to preserve is, again, this mystical consciousness. All right? So these were all things that we were quite interested in interrogating as part of the project with Dietmar's team. So one of the sort of starting mechanisms that we developed was biosynthetic systems, and this was an exhibition that was actually displayed just across the road at the block. And that was with a big sort of research team starting to look and explore some of those implications. So thinking around, well, if you could magically, and again I don't believe that you could preserve a consciousness in a digital format, but if you could, and you could transplant it into another body, but it's a different body, maybe a younger body with a different gut flora, with different physical capabilities, is it still the same person? Is it still the same person or the same consciousness if you transfer it into a digital interface that again has very different embodied kind of connections into the world? Or is that again different? What happens when you actually merge biotechnology and artificial intelligence and you create something that evolves, that has its own agency, that it's not just static? Is it the same person then, if it becomes different over time? So these are sort of some of the questions that we wanted to interrogate, and we did that through creating a series of works that basically operated to stimulate debate. So one of the things that we included were a live sort of 3D printing demonstration that also showcased the scaffolds. Part of the reasoning behind that is that people do have this perception that is again around this hype that you can 3D print an organ now. That we're, like maybe 2 years away and then suddenly we're going to be having all these organs printed everywhere, but we're nowhere near that, So it was really trying to showcase that, yes, the technology is evolving rapidly, but we're very far off, and the body is a much more complex space than we are often led to believe. So it was kind of showcasing, well, this is where we're currently at in terms of the height of 3D printing and what's going on in the labs. So that was sort of set up, but it was also coupled with some of these early engagements. And this was work that was developed with Bill Hart, and you can see there my face, which was kind of mapped with a 3D system. I also wrote my handwriting, and I started to write a kind of narrative that was around imagining what it would be like to be myself, but as a biotechnological kind of AI entity. So I sort of started to write this narrative. And then Bill created this writing machine here, which actually draws on programmed neural networks to recombine and to recreate that narrative in my handwriting, but that it actually changes and evolves over time as well. So it sort of was about this idea of, well, what happens if you do create a machine, you upload your consciousness and it does evolve? What might it start to lament? What might it start to say? And there was a kind of pathos to the way that the words sort of started to combine, and sometimes it would make sort of sense and then it would deteriorate and then it would be like nothing, 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 and just repeat and sort of... So it was kind of quite a provocative work, I think, and that was combined as well with these video works of my kind of face that would sort of split apart and move, and was sort of around again this kind of disembodied entity that was now in a completely different environment. So along with that, we also were working with some of the Electrospun scaffolds that were produced in the lab. So here you can see one of the scaffolds that Felix Wunner, who will be actually presenting a bit later today, and I'm sure he'll tell you about his PhD project. So we were fortunate enough to work with one of the electrospinning devices that he had developed, but we actually redesigned it, well, he redesigned it to actually also integrate my handwriting and then to seed cells onto that so that they would grow. Here you can see sort of one of the scaffold at high resolution with the cells growing over it. And they were actually tagged with the green fluorescent protein so that they would fluoresce under UV light, because that would allow us to actually track the movement of the cells. And that movement was then translated into the development of an STL file, so a three-dimensional file which you could then print. So it was also around, well, looking at cells as also having agency, that they're not these inert materials, but they also, you can't always control and predict how organisms are going to progress, especially when you have evolutionary pathways, whether it's a synthetic kind of evolutionary system or an organic evolutionary system. There's always an element of uncertainty. And so it was around allowing those cells to actually have agency on the three-dimensional structures that they're producing. But again, these are all sort of early ideas that we were investigating. And this also were developed because we were interested in seeing what the scientists were really keen to develop, because it also challenged the limits of the technology. So it was also around, is this an interesting problem for a PhD student to work on from a technical perspective and also from a kind of philosophical perspective? So we developed also this biomonument, and that sort of started to house a resin printer that would print the three-dimensional structure created by the cells. And then in the top there was a little holographic projection of the cells moving through the structure as well, with text that was generated. So it was again all of these interconnected elements that started to encourage the viewers to consider some of the implications and to kind of become provocations for further dialogue. We also showed aspects of that work at the National Science Week event, the Festival of Bright Ideas. What was really fantastic about that, there you can see Felix and myself talking to literally thousands of school children. And so this is also where, as an artist, what I'm always interested in is the way that art can actually have multiple functions. But it's not about here, you know, if you looked at this, you could go, is it about science, communication and PR? Absolutely not. The idea is about critical engagement. So you actually present students with a range of options and you get them to actually think critically about the technologies. That's the purpose that I kind of see in connecting with the public and also with school children, that it's about encouraging that critical debate. So for future iterations, we're hoping to develop this project further. And again, looking at the connections to the 2045 Avatar project, looking at notions around robotic bodies, interconnections in that way. So thinking about how you might connect the programmed neural networks, but to actual neural networks, and then actually develop, and that you can do that with using a multi-electrode array where you actually seed the cells on there so that you can send and receive electrical signals. I've got a good friend of mine who is based at Symbiotica, which is a dedicated art and science lab in Perth, which Elizabeth will talk more about. But actually, so the resources are there in being able to actually refine that technology to work with a robotic body, and that's where we're kind of interested in that interaction between programmed artificial intelligence and then also real neurons, which are again kind of seen as the brain, even though again, very reductionist viewpoint. But it's about that critical interrogation and drawing on the work that a lot of artists have done previously. So artists like Ken Ronaldo, who's done work with fish that are controlling these robotic bodies, and then also people like Stella. So again, this comes to, well, why, okay? And I think that's going to be a question that you'll see Elizabeth talking probably a lot more about. But again, for me, as I've sort of alluded to, it's about provocation, that art actually, the role of artists is not to provide solutions. This is something that Oren and Yona continually stress, who are leaders in the field of art and science. And it's about debate, but also critical interrogation. And these are just some of the projects that are really pioneering works within the realm of art and science. Joe Davis, who was one of the first people back in 1986 to 2000 to explore how you might encode information into DNA. Eduardo Katz, who created the first transgenic organism, so a bunny that was engineered with the green fluorescent protein, intended to be a pet. But it was again about a provocation. What's happened since then? We actually do have glowfish, which are transgenic pets. So it's almost like preempting where the technology might end up in a kind of speculative way. Paul Vanus, who did his latent figure protocol, you can look at it there, repurposing technologies to actually draw attention to bigger implications around copyright of biological organisms, tissue culture and art project in there, Disembodied Cuisine, which Elizabeth I'm sure will touch on more, but really interrogating this idea of lab-grown meat. I would just like to add, corn already exists. It has texture like meat. It's a mycoprotein. Of course, that puts people off because it's fungus. So again, it's around how do we shift the kind of cultural attitude where we don't have to have meat. You know, to me, it's kind of quite bizarre. And then, of course, Stella, who had his ear on the arm, which was looking at embedding the scaffold in another place. So, and you'll see a lot of artists will actually start thinking about, well, why not replicate the same body? Why not extend it? Why not actually allow other avenues of thinking around embodiment and what constitutes the normal body or the ideal body? So it's again that critical interrogation. I'd also just like to mention that what you'll see as well is the growing interest in the area of art and science. So the Science Gallery Network is something that you can see was originally established in Dublin, but has since spawned multiple offshoots. So we've got Dublin, London, Melbourne, Bengaluru, Venice and Detroit. And you can see again it's about provoking the debate with the wider public around things like automation, climate change, synthetic biology. And one of their latest call-outs was spare parts, and that's one of the call-outs where we've put in an application which we're hoping will allow us to showcase our project, looking at, again, this idea of immortality and, again, the whole body being seen as a spare part. So allowing us to actually move into a space that allows the public to connect with the realities of what's going into the lab, but also to critically interrogate all of those speculations around where we might go into the future. So I will just sort of mention that this sort of forms part of an ongoing series of explorations. Other areas that I'm interested in as well, which all kind of connect into this idea of a distributed self, how you might actually proliferate your identity through multiple avenues, and through both digital and biotech avenues. So looking at, well, what might happen if we have non-human offspring? What might happen if instead of procreating via children, I can actually infect someone with a virus that will replicate fragments of my DNA? What might happen if, so it's asking these sort of questions to probe, well, what are we comfortable with? What are we fearful of? So it's again sparking that debate. And I will finish there, which hopefully leaves me at around about the right time. Elizabeth is very impressed because sometimes I do presentations where I'll go, I have 62 slides for 15 minutes. But I'd also just like to say, thank you to all the people that I work with. When I talk about a lot of my projects, they are collaborative. So I do work with a whole bunch of really amazing people, including the Tissue Repair and Regeneration Group, the Centre for Regenerative Medicine, but also most recently with people from Tasmania, so from the Creative Exchange Institute, and also Bill Hart, who's from the School of Creative Art. So really amazing people that all have excellent skills. Thank you to them. And I will end there.
Okay, thanks everyone. Thanks so much, Matthew, for inviting me to participate today. It's been really interesting to hear everyone else's presentations, and I've been a big sponge learning a lot from this. So to begin with, let me explain the kind of context from which I am approaching the issues, both of biofabrication and bioprinting. So my approach is one of cultural analysis. I'm a cultural theorist. I come from a history of science and a contemporary science and technology perspective. So what I'm going to do in my presentation is just take a couple of minutes at the start to explain the kind of frameworks, the theoretical frameworks with which people like me approach these kinds of issues, and then I will focus specifically on biofabrication and bioprinting in particular, but you'll have more of a sense of the sort of areas that I'm coming from. So these are the sorts of questions that people like me ask when they come across technologies, new technologies like bioprinting. As a historian of science, the first thing that I look to are what are the historical antecedents of these kinds of technologies, which means looking at the history of the technologies themselves, but also the cultural systems, the economic systems, who's paying for these developments, the legal systems, the professional networks, institutional contexts, and so on, in which they're developed. The next thing that we're likely to look at are philosophical. So that means what kinds of ideas are these sorts of technologies emerging out of, what sustains them, but also what kinds of questions do they pose for people in contemporary societies. The other element is aesthetic. What is the role of demonstration, display, and visual images in the development and circulation in general publics of these kinds of technologies and their products as well? And in particular, to what extent is the visual culture of science and technology constitutive of scientific and technological knowledge rather than just illustrative of it? And then finally, and what brings us all together, are the cultural questions. So how do members of the general public make sense of these new technologies? How do they understand them? When they go to an art exhibition, like one like Svenja's, how do they engage with works like that? When they hear about this kind of work in the media, are they able to critically engage with the kinds of comments that are made? So what I want to do just briefly is to just unpack a little bit what it means, what it looks like, what it gives you in terms of understanding histories of science and medicine, to ask these sorts of questions of the history of science. So I'm going to use as a first example, because I think it's really appropriate to the background of developments in bioprinting, my second book, which was on public anatomical displays. Now, the significance of this for our purpose is that at the time that a lot of the public anatomical displays that I focus on were happening in the Victorian period, it was a time in which anatomy was an extremely disreputable area of medicine. So in the 1820s, for instance, there were public riots against anatomy schools, including very violent riots when people went to the head of the Edinburgh Anatomy School's house and stoned it. So there was a lot of angry public reaction to the practice of anatomy because it was considered a violation of biology. I think it's interesting to question whether this might inform how people respond to manipulations of biology with contemporary technologies. What happened was that there were a series of legal changes that went as a result of these public riots that were basically the formation of the Anatomy Act in the UK in 1828. The interesting thing, especially because so many people here are focusing on legal contexts and histories, is that it was not law that changed the status of anatomy in the UK in the 1820s. It was public opinion that changed the laws, that changed the status of anatomy. So it's interesting to be aware that it's not just about public policy, it's about public opinion as well. So my interest in this book was in the fact that it was through public displays of anatomical models and specimens that there was a transformation of public opinion that allowed a legal transformation that allowed the contemporary sphere of medical research in anatomy to take place. So it's not a history that tends to be told in histories of science because the key player in that history is the site of public spectacles and fun fairs and sideshows, and that doesn't tend to be where people think the history of science takes place. So just quickly before I move on, this is my third book, which has just come out this week actually, so it's hot off the presses. And in this book, we're looking at the history of the idea of normality. Now, the significance of this is that we are looking in this book at the history by which a term that starts off in medicine primarily moves into the popular sphere very late in the middle of the 20th century. So the term normal starts in medicine. It refers to normal anatomy or the normal state. And these just mean the usual organisation of the organs or a state of overall health. So normal in a medical context basically means healthy. It's only at the end of the 19th century that the word normal becomes a term that's used in statistics, where it becomes the word normal used in the phrase normal distribution, which most people would know basically describes the bell curve, that most data will cluster in the middle of the range. So our interest was, how did this word that was used in medicine just as a term of scientific jargon, and then a word in statistics just as a mathematical term, come to explode in the public sphere in the middle of the 20th century and become the popular word normal that is used, on an everyday basis now. Again, interestingly, the agent there, the catalyst for this, are public exhibitions. They're public health exhibitions in particular in the middle of the 20th century around the idea of the normal body that were on display in popular places, in World's Fair, all in the middle of the 20th century. And suddenly the word loses its scientific specificity, and the idea of the healthy and the statistically average merge, and it becomes a word that's used in popular discourse. But again, the agent of change is public exhibitions and public spectacles. It's not the dissemination of scientific knowledge per se. So this is the framework with which I am looking at or approaching my current work, which was nicely mentioned in the introduction to this panel. I'm just starting a future fellowship. I'm just a couple of months into it, so I'm interested in all feedback on it. And it is looking at a cultural history of the experiment and practices of experimentation, focusing particularly on art science collaborations. So my hypothesis is both that collaborations between the arts and sciences play a crucial role in this history, but also that our tendency to think of these as separate fields of research is actually a very contemporary one. In terms of the kind of philosophical framework of the project, my interest is in the way that the idea of the experiment has become so dominant that not only it starts in the sciences in the 18th century, but then it moves into all other cultural domains. So, you can see from Svenja's work as well that cutting edge work in the arts is also very experimental. So I'm interested in the kinds of forms of knowledge production that come out of this and why it becomes such a dominant one. So I want to look at that by looking at a couple of examples of biofabrication and bioprinting. And I want to start with this example of experimental or speculative biology. Have most of you seen this image before? It's quite a well-known one. So this is the mouse with an ear on its back, and it's the first successful example of tissue engineering. So this is a human ear on the side of a laboratory mouse. It's not grafted on, it's actually grown onto the mouse. That's what makes it such an important image. The image was first circulated to the public via the BBC on a popular science broadcast, and it's always cited as the first successful example of tissue engineering. So a couple of years ago, at the end of 2015, I co-curated an art exhibition on the ear mouse with the colleagues that Svenja has already mentioned, Aron Katz and Ian Atzer, who runs Symbiotica at UWA, who did their own training as biological artists in the Vacanti lab. So they were learning tissue engineering as an art practice at the same time that tissue engineering was developing as a medical practice. So what we were interested in was the cultural impact of the ear mouse. And so we were interested in well-known artists like Patricia Piccinini, who did this series of protein lattice representing ear mice. and also Stella, whose image we have already seen today with his third ear as well, and the way that the ear mouse had sort of moved into popular culture. But we were also interested to show that the original image of the ear mouse was itself a very spectacular image. It was represented as a kind of spectacle, and we were interested in the role of spectacle in relation to speculative biology in gaining prestige, in gaining a reputation for the scientists, in terms of getting funding for their work and so on. So we didn't want to see the spectacle of the artists and the spectacle of the scientists as operating in separate fields. We saw them as operating in the same place. So let me show you another example of the work of Aron Katzen and Ian Atzer that relates more directly to bioprinting and the kinds of commentaries that they make on this. So this piece is called Mechanism of Life, after Stéphane Leduc. Does everyone know who Stéphane Leduc is? He is one of the originators, 1911, mind you, of synthetic biology as a field, so a kind of proto-synthetic biologist. He wrote a book originally in French. The translation was in 1911, and in English it's called Mechanism of Life. And so his idea was, he was a chemist, and his idea was to experiment with combinations of chemicals that would produce semi-living systems that would imitate living systems and living processes. And so people often say now that he's the originator of synthetic biology. So in this piece, it's an attempt to reflect on, it's a reflection on Stéphane Leduc's work. And I just want to read out to you the artist statement for this, because they will explain what it is that they're doing in this piece. And I'm quoting them because they're quite critical of bioprinting, so I'm taking a little, allowing them to say it in their own words. They say, in this piece, we reenact one of the simplest protocell protocols offered by Leduc, working with the diffusion of two concentrations of solutions that create transitory cell-like droplets. The droplets resemble cells, they're not actually cells, with membrane and nuclei, and they last for a few moments before succumbing to entropy and dissolving into a murky liquid, much like life. And let's just go back to the, this is the cover of Leduc's work as well. So you can see he includes a lot of artworks in his work too. And here is a close-up of the piece. They say the protocol is automated using another hype technology, three-dimensional 3D printing. There is much discussion about 3D printing technology as the next industrial revolution, and we've heard some of that today. Something that parallels the assembly line of Fordism at the time Leduc was working on the mechanism of life. The promise of 3D printing technology, they say, is at its core based on information transfer as the business model. The focus is on instructions data as the currency, while the materiality is merely an optional manifestation. And the quote that Matthew showed from Westworld, where the importance was on the intellectual property rather than the organisms produced, I think is another articulation of that. So to continue, they say, this very seductive scenario of printing life from scratch is played off in this work against the unstable, uncontrollable and transient nature of the protocell droplets as material. And I think Svenja touched on this a little bit as well. In a time when the idea of creating synthetic life is at the forefront, it's important, they say, to culturally probe the current and past approaches to the idea of the mechanism of life. The printed protocells are unstable and temporary. They take on forms that appear organic and then disappear. More than a proof of the mechanism of life, they are a suggestion for a humble approach to the question of what life is and how far we are willing to make it a raw material for our own ends. So that's a very critical account of the role of bioprinting technologies and how they see their own work in relation to that. And this is a big part of Aron and Ianette's work as a whole, is to use artworks to question the hype surrounding a lot of these technologies. So because Hope touched on this earlier, I thought I would just throw in a couple of slides of another example of this work from 2007. 2003. This is Disembodied Cuisine. It was a project to manufacture synthetic meat. The project is critical of the assumption that, because they show the manufacture of the meat, the project is critical of the idea that it's a victimless process for the reasons that Hope mentioned in her presentation, that in fact it's engineered using animal products, so it's not victimless at all. But also one of their points is that this is the size of what it was that they could make in 2003. It's tiny, for one thing. So the question of scale that Hope was mentioning is a really important one. Also, the texture was so awful that when people ate the synthetic meat, most of them spat it out, even though they knew that it had cost a fortune to make. And so the artists collected all the spat out, bits of meat. And then they had a follow-up exhibition called The Remains of Disembodied Cuisine. And again, it's a comment on the disparity between the hype of these kinds of technologies and what can actually be produced. So you'll imagine their amazement, the artists, when 10 years later this man, Mark Post, the scientist, released his notorious Frankham Burger, eaten by journalists of the BBC, at, again, another very spectacular PR launch for this. And they were critical of this for a couple of reasons. One is, you might notice, this is 10 years later and it was claimed to be the first example of lab-grown meat that was eaten, and they're like, well, actually, we did that 10 years ago. And the other thing is their concern about the way this is being presented to the public. This patty here, cost £250,000 to produce, for one thing. And also, this isn't all the artificial meat here. This is filled with filler. And the question of how much burger helper went into the burger and how much is actually synthetic meat is not in any of the information that's publicly available about this, but it's as much as 90%. That's a very big burger. So again, their work is critical of the kinds of claims that are made of this. But I want to turn now to another example that's directly relevant to bioprinting. And again, it's an artist that Svenja has already mentioned, which is this man, Joe Davis, who's one of the pioneers of this field. And he recently began a project called Malus Ecclesia. Now, if you don't have Latin, malus is the word for bad or evil in Latin, but it's also the word for all genus of apple trees. And the word ecclesia often obviously refers to church, but also to George Church, who's a professor of synthetic biology at both Harvard and MIT, and Joe Davis is collaborating with him in this project. Now, for this project, what Davis is doing is he is inserting the entirety of English language Wikipedia into a strain of 4,000-year-old apple to create a literal living tree of knowledge. So I'm interested in this project because it tells us something about the relationship between the biological and language as well, that I think is interesting to think about when we're talking about bioprinting. To what extent is bioprinting printing, for instance, we might ask. So the project's still ongoing, and we don't have any actual apples yet, but it's a commentary on the sort of Judeo-Christian, construction of knowledge, but it's also a sort of spectacular giving in to the temptation, that's at the heart of that story, because he's actually re-engineering the central metaphor or medium of the story. So if in the Bible humans are word made flesh, Davis is proposing to take flesh and turn it into words by making these wiki apples that are made out of text. Now, as I said, we don't have any actual apples yet, but he has been talking about the process at a number of recent conferences and had some installations on them. So here are some sort of renderings of paper versions of this. And you will notice that he has the DNA lettering, around the side here of the books that look like apples, because, of course, I probably don't need to explain to people in this room, as I do when I'm explaining this to people from a humanities background. But the way this process works is that you translate English words in this case from Wikipedia using the 4 letters of DNA, and he's actually using for this a carrier bacteria. So he's actually doing the translation of carrier bacteria that will then be implanted within the apples themselves, and the bacteria is benign, so it won't affect the genetic makeup of the apple itself. It will, however, be illegal to eat the apples on FDA grounds, so no one can give in to temptation in this grade. So what I'm interested in with a project like Davis's is it's also a commentary on the role of language and its status within the history of science as well, and within the history of knowledge, within Judeo-Christian concepts of knowledge as well. It's only because we conceptualise DNA as being made up of something we're calling letters that you can even do this process at all. And so his work sort of looks at the role of linguistic metaphors and ideas within both the history of Western knowledge but also within contemporary science practice as well. So let me just finish off by showing you a couple of examples from some other colleagues that Svenja has touched on, the work of Stella, who I don't know if you know, here's some more images of Davis's work, but the artist Stella recently opened, it's now in the process of closing, a lab called the Alternate Anatomies Lab at Curtin University, which is also using 3D printing technologies. Again, it's a kind of commentary on the interface with the bio. They're not doing bioprinting there. But here are a couple of projects that he's been working on. So this is a robot fish that one of his PhD students has been developing. You'll notice that all of their projects are using mechanical components for actual movement in them and to simulate life. And all the shells of the pieces are 3D printed in the lab. They have their own very good 3D printer. So this is a robot fish, and it uses the robotic elements so that it's Bluetooth connected as well, so that it can be remotely operated, and it has the 3D printed shelf. But they're also experimenting with a number of pieces that are a medical interface as well. So this is the ambidextrous arm. The idea is it can operate as either a left or a right arm, and it's sort of, all the fingers are double jointed. Again, it's using a series of motion sensors, for the actual movement of the piece. So it can be remote controlled as well. And the fingers and the hand itself are 3D printed. And then I think this one's really interesting because the lab was moving a lot in this direction, doing sort of boutique-made prosthetic pieces of various kinds and often at the request of people, coming to them. So in this piece, again, there are sensor technologies used, and the idea is it's for patients who might have some sort of, hand entropy or paralysis of the hands. People who have had strokes are a good example of this. And the idea is that it can be remotely controlled as a form of, like a sort of form of physiotherapy, if you like, with increasing control given to the patient themselves. But the actual sort of, orthosis that you can see here, the kind of harness, is 3D printed as well. So they've been looking at the fact that you can work with much more specific measurements. So they're working very much at the interface there. So I wanted to show this as a closing example because I wanted to show the kind of range of people who are working in collaboration, artists who are working in collaboration with people working in scientific research in the sort of bioprinting and larger spheres, are doing it in a range of ways. Some of these pieces are collaborative in the sense that we might understand collaborative in that they're joint projects, but some of the collaboration takes the form of a critical engagement as well, and I think that those works are important too. So just to sort of conclude with, what I think is the purpose of these kinds of works is that as Svenja has so eloquently said, they're provocations as artworks. They don't provide a didactic account of what these sorts of technologies are doing. They provide a space in which questions can be asked about them, and I think this is really important because I was at the opening of Svenja's exhibition that she showed all those images of just across the way, and I had a bunch of colleagues from UQ with me, and they were following me around asking me to explain technologically the artworks to them. It's like, well, actually, I'm not the person, I'm from the humanities, I can't explain them to begin with, you can ask Svenja. But also I kept saying to them, but is this how you respond to other artworks? Do you feel like you need to understand the technique by which the brushstrokes will put on? Why do you feel like that with these, artworks? What I think is important about these works is they provide a space of encounter for the general public in order to have some way to interact with these technologies. And ideally, they do it in a way that provides a space for critical engagement. Because I can tell you as a non-scientist in this room, what happens when people from the general public read popular popular media accounts of these technologies is that they don't understand the technology, so they don't understand what they're reading, so they believe everything. And if there's an illustration, even more to the point, they will, oh, there's an image, so it must be true. So these kinds of artworks provide a space that is a bit more familiar to people, where they can go in, see the technologies that they're often just hearing about, find a way to critically engage with those. And I think for the scientists in the room, for people coming from legal backgrounds, it's important to all of us that people from the general public have a way to critically engage rather than passively absorb the new technologies that are coming through. Because as I explained with my first example of the history of anatomy, it's in fact not just the technological developments or the legal frameworks that will enable these kinds of technologies to find an application in the contemporary sphere. It's actually public opinion and public acceptance of these technologies, which means understanding them, that will make the space for that to happen. Thank you.
Thank you both, Svenja and Elizabeth. That was fascinating. I really like that idea of art as the provocateur or questioner or way of critically engaging with new technologies, because I think that's a lot of what we try to do in law as well, but in a far less entertaining way. Questions? Yes.
Thank you both. I suppose this is more to you, Elizabeth. Rather than actually the artwork being a space to facilitate the acceptance of the technologies, how do we influence, how does the public be involved in how society develops using these technologies? How can we assist in that process? Because when you look at the technologies, like for example, I didn't watch Westworld because I just went, that's way too rapey for me. The future is extreme. Yeah, it's really shit, and I don't want to, just look at the news if you want that. But how can we somehow use art to create spaces that can influence how the technology is used in the future? So ultimately we use technology, I would argue, for creating better rather than the Westworld perspective. Not that that's real.
Yeah, I mean, I don't think that there's a kind of a simple causal chain in which you can have an art exhibition and that's going to transform some public opinion and that's going to allow to. So I'm more interested in, I guess maybe a less instrumental approach to that kind of thinking. So for instance, one of the questions that people like me would ask of something like Westworld, for instance, is what are the kinds of narratives, underpinning the use of this technology and the way that it's, played out in future spaces or likely to. So, I mean, Westworld, you mentioned the rapiness of Westworld. Westworld is one example of this. But when the new Blade Runner came out as well, there was a really strong gendered reaction to its vision of the future. For instance, why are all the sex robots female, for instance? Where are our boy bots, the feminists wanted to know. So what interests me is not, is that a plausible, representation of the future? Is what are the narratives, underpinning this? And I think that one of the things that spaces like art exhibition spaces can provide is a way to provide a cultural language and to provide conceptual frameworks within which brand new, very unfamiliar things can start to literally make sense to people who don't make them at all. What the mechanisms of that might be for a general take-up, I think is a much broader and more complicated set of questions, but I think it's the starting point that people need to, like a lot of the questions that people have been asking in the room today, like to what extent are the sort of conceptual frameworks that we're using in the questions that we're asking really Eurocentric? How might those play out in other cultures? These are really big sorts of questions. To what extent are we influenced by different frameworks of religious thinking or ideas about gender or ideas about embodiment or ableist ideas or, whatever it might be? It's a space to ask questions, and I think until sort of general publics can have a way to even start to engage with these technologies, I think that is one of the barriers, to the uptake of them. But I don't have a sort of list of 3 steps that would then turn those into technologies that can be easily applied. Svenja probably has other thoughts on that?
No, I think I agree with you, and I think that one of the things that I was just thinking when you were talking about that is also that what's so great about coming together in this forum is that I think that what also is important is that you have perspectives from other disciplinary areas that can shed light and ask different questions so that you're actually engaging in the debate. And I think that that's one of the things that I'm really interested in working with scientists and also with people like Elizabeth who come from a humanities background, because the questions will all be really different and that it adds into that critical interrogation. And I don't think that it's just the role of an artist or just the role of a cultural theorist. I think it's very much a role that we can all take up from the position of our own disciplinary expertise to actually go, well, you know, the way you're representing, you know, the science is inaccurate because these are some of the limitations. Well, you know, when you look at this, you know, so that there is that dialogue, and I think that that's where we can actually make a lot of difference as well and encourage public debate, because it means that they're actually seeing a multitude of positions without necessarily saying, but this is the right position, but actually going, well, here are a range of debates, engage in this. Where do you sit? What is your position?
See, that leads into a question I had, which is around, so Elizabeth mentioned that we often think of science and art in very siloed ways, and even in our universities they're very siloed. But both of you work at the nexus of science and art, or science and cultural studies. And I was curious about your own personal experiences working in that interdisciplinary space with scientists and other people speaking different languages. How hard was that to kind of overcome any language barriers or other barriers?
Did you want to start with that?
I mean, this is something that we just recently had, the Critical Connection Symposium, that was really starting to address, well, what are the barriers that are in place in terms of, first of all, allowing for that cross-disciplinary communication, and then also allowing for interdisciplinary engagement even at lower levels, at an education kind of level. And you're finding that it's a massive issue, and part of it is because we do have these siloed structures, and it takes a long time to build relationships across disciplines, to build trust, to have that articulation of language, and a lot of the structures that we have are not really conducive to facilitating that sort of engagement. So it's a difficult one, because I think that at the moment you have people that are really passionate about working in that field, and I also don't think everyone needs to. I think that we also do need that disciplinary expertise where you have experts that are, you know, really understand their field, and then you might have people that work at the periphery as well. But I do think it's really tricky to facilitate that, and that's one of the things that I'm really passionate about, looking at how do we continue to facilitate and allow that to occur, because I don't think it's an easy one, and a lot of it is around time, and particularly within a research environment, you just don't have time to meet people, to find the people that you want to collaborate with, where there is that chemistry. It's all around that kind of build up that takes time, and it's not currently allowed for, so there's a lot of those sort of hindrances which need more unpacking. But
yeah, you know, I mean, it's an interesting experience if you haven't spent any time in scientific spaces and in laboratories to go and actually do some cultural anthropology, you know, so for someone like me, you know, who hasn't grown up in those kind of environments, and it's a good way to see behind some of the claims that are made in popular contexts about this. So again, the synthetic meat example is one that always sticks in my mind, because the first time I spent any time in a lab, and Svenja in fact was one of the people who took me, you know, for some of my first tours through labs, the thing that's really striking is how much waste is developed in a lab. You're always like taking off, you know, gloves, and you're taking everything off and you're throwing everything away. So, oh my goodness, there's a lot. You can't recycle anything, you know. So then when you see claims about how clean these things being made in labs are, you're like, you know what, like I would like to quantify, you know, the amount of waste. So I think it's helpful in terms of, if you're like me and you do work in the history of science, it's helpful to have some experience, you know, in contemporary science spaces. But I have to tell you as well, as someone who's a historian of science, that everything people in the room here are doing, those of you who are like scientists rather on the legal side of things, if you're working with technology, go back beyond the beginning of the 19th century and you were all artists. You were not scientists. So in the definition of, before there was science, there was natural philosophy and natural history. And I'm talking till the end of the 18th century here in the scheme of things, it's not that long ago. Anyone who worked with technology was working with techne, it's an art. And so the real, natural philosophers and scientists of the 18th century have all these denigrating comments to make about technologists, and they call them the scientific arts, they were called, up until, like the sort of 1830s, they were all referred to as the scientific arts. And they would say, people like Buffon, very well-known natural philosopher of the 18th century, said they're the scaffolding upon science, they're not science itself. So in fact, technology developments, all these biotechnology developments, were the arts, until quite recently. So I find that kind of perspective quite helpful as well.
Mia?
Thanks so much. Those talks were absolutely fascinating. And I guess I'm speaking from a scientist in the room rather than the creative side. I think when we look at a lot of these images and we talk about the hype, which of course is always sort of hinged upon with a lot of the scientific stories, we've always got to remember that most of the time scientists are sort of coming up with these new ideas and these new technologies, and it's when the media steps in that things start to get hyped up. Not many scientists would sit there and say, hey, I'm going to show you this tissue engineered meat in a hamburger in front of a camera. That's often influenced by your team within a university or within an industry who want you to sort of, hype to a certain level the audience. And oftentimes it is misrepresented, and it can have a serious impact on the scientific research progress. Often, every group has sort of had that happen. So we have to be really careful with our, our communication, and we have to learn a lot from how to communicate the science to have enough hype to be interesting, but not too much to think that you can grow ears on mice, because you can't. So just to clarify that ear, the seminal science paper, the ear grown on the mouse, it wasn't grown on the mouse. It was printed or created outside of a mouse, and then some cells were put onto there, and then it was simply slotted under the skin of the mouse to look like an ear. So it wasn't grown on a mouse at all. And so I think we have to be really careful how we tell that story, because even now people believe you can grow ears really easily, but actually to those of us who are trying to do that, it's really complicated, and there's a lot of things we still need to overcome. And so we've got to be really careful with how we, put our science forward, but also how it gets communicated as well. And with the bioprinting, and so talking about the gels and the cell structures. So the cells you were talking about, it's confusing because a cell is a living organism, but also you talk about cellular structures within a material. And so when you say that they dissolve away and the membrane disappears, that's a material property, it's not a cell property. But you can actually combine both of those, and you can put cells into a hydrogel, and you can bioprint human cells into a structure to create a tissue. So that is possible, and a lot of groups are doing that already. So I think this is a great exercise in sort of seeing how these stories, because you present them so beautifully and much more creatively than certainly we do, and it's getting that good balance, which is why it's so nice to see everyone coming together today to talk about that interface, because I think it's really important. So I've certainly learned an awful lot watching your presentation, so thank you.
Thank you so much for that. I think that's a really important comment as well. Just on the mechanism of life, piece that I showed, the bioprinting piece, that was not. They weren't using any, living materials in that at all. It was a representation of that. Although interestingly, that's not clear. When you see when you see the didactics of it. But I can tell you the way the Vacanti brothers themselves tell the story of the mouse with an ear on the back is that it's an ear grown on a mouse, and they're telling that story now. They're also continuing to do the project. So now they're growing, developing, grafting, ears on goats at the moment. And certainly they continue to tell the story that way. So these aren't just humanities people's, misrepresentations or public media representations. There's a point at which they're the narratives, told in this case by the Vacantis themselves. And that's part of what, people like Symbiotica are drawing attention to, the role of narratives, in the way that the history of science is understood and contemporary scientific developments are understood. I think think your point about the role of media in hype is a really important one as well. And I think it's very important for people as ideas and development move across different cultural domains. So they might, one of the things that Svenja and I have noticed is they don't always start in scientific laboratories. Sometimes they in fact start in art, experiments. Wherever they start, then they move across all these cultural domains. And one of those is the media. And then you do get these sorts of translations, of stories into languages that a general public can understand, which might look like these hyped up claims. I think that's true, but I do think it's true as well that members of the general public often get accounts of scientific developments that make the nature of those developments unclear to them. So, one of the phrases that always makes me smile is that you hear this, an account of a new development of, technology or whatever it might be, and then inevitably seems to come the frame, and so we anticipate that within 10 years. And now whenever I see the phrase 10 years, I kind of giggle. It's like it's the plausible deniability, framework that it's just soon enough to allow some more funding, and attention to the work. We all have to have public engagement and impact, going on. But it's just far enough away that people might have forgotten that you might make that claim. So.
Well, I think we're at afternoon tea time, so please join me in thanking again Svenja and Elizabeth.